told you to come and eat and hang out, so please do. And we have a Q&A with him tomorrow at noon, which is an unusual time for this, but it's noon in LSP 207. Please send your questions ahead of time. You can do it on the link to the seminar announcement. And that's it. I'll let you go.
All right. Hello. How's that going? My voice doesn't carry that well, so I'm always happy to see the microphone, but hopefully y'all can hear me in the back there. Well, thank you, Karen, for an awesome introduction. It's always really fun to kind of hear what people think of my work, and so it's really fascinating to know that it resonated with the community here. I had some very fantastic conversations with grad students and faculty here, and so it's been a really awesome visit so far. As Karen mentioned, I'm from Houston right now, which was like 80 degrees, and so it's a little bit bizarre getting off the plane. It's like 30 degrees, and so it's like a 50-degree difference there, but I guess that's expected for this time of the year here. But a beautiful landscape, and it reminds me a lot of my work in Iceland with just mountains in the backdrop there. So with that being said, I consider myself a soil ecologist by training. I'm really interested in anything and everything that lives in the soil, so really thinking about soil biodiversity, and really in the context of how it relates to ecosystem functioning. So think about nitrogen and carbon cycling, but particularly in the context of global change. And so what I like to really think about is, these organisms in the soil all have a role, and it can really influence how nutrients are recycled. But thinking about how invasive plants can change those interactions, or how urbanization can impact that, or how climate change. And so broadly speaking, I consider myself a global change ecologist, and all with the idea of trying to link soil biodiversity to ecosystem function, in that case there. And so with that in mind, the title of my talk today is going to be Unraveling Soil Community Dynamics in the Era of Global Change. So, as humans, we kind of stand upright, as we sort of walk through, any forest or grassland for that matter. We are naturally sort of drawn to the things that occur above ground. And as you can see here, we got, beech maple forest, so you got some beech and maple sort of in the overstory, but you also have a diversity of plants that occur in the understory. And we kind of naturally drawn to the greenness and all the sort of biomass above ground. However, if you were to take a moment and actually think about this, 80 to 90% of what you see above ground was recycled through the soil ecosystem at some point in time. And so one can envision that everything you see above ground is actually fueled by all the organisms and all the interactions that occur below ground that allows for what you see above ground to actually persist in that case there. And so one can envision then that the soil ecosystem is an incredibly important ecosystem that we should learn more about to really understand how it drives all the interactions and plants that we see above ground. So as we sort of continue to walk into, this beech maple forest, kind of depending on the time of the year, what you actually see is this very healthy leaf litter layer here. And oftentimes people kind of take for granted all the different organisms that live below ground. But if you could just take a moment and just kind of look at all the various groups that you see here, you will understand it's an incredibly diverse system. And in fact, there's a sort of an instant classic that came out last year in PNAS by Mark Anthony, which actually showed that about roughly 60% of all the species that occur terrestrially occur below ground. And so you can think about fungi, bacteria, all the invertebrates, encompass a huge amount of diversity in that case here. And the point of this slide is actually to highlight that diversity. And this is just going to kind of go through some of the really important groups that I really think about a lot. So with that in mind, as you're sort of walking through this forest, you take a moment and look down. If you were to actually recognize all the organisms that exist there, you would notice that there's some bacteria. Admittedly, you can't see bacteria with the eye, but you can kind of sort of envision that there's going to be a whole host of different bacteria that occur there. They are incredibly abundant, incredibly diverse, and they also do some really important activities such as liberating organic matter, making that organic matter into plant available forms that then allows for plants to uptake from the environment. We have fungi, an incredible group that we still don't know a whole lot about, but there are certain categories that are incredibly important. Some of them sort of form mutualistic interactions with plants. where plants get sort of nutrient absorption and moisture from the environment, and the fungi get carbon from the plants and sort of form these critical mutualisms below ground. We also have saprophytic fungi that actually can be free living. It can also break down organic matter and release that into the environment for plants to uptake. And these are actually really important organisms that can fuel a whole sort of ecosystem of other organisms, such as springtails, which I would get to in a short bit here. So just these two groups alone, they kind of make up a huge amount of diversity that happens in the soil, and they, as I alluded to, have really important functions in that case. We have predators, which are centipedes. They eat other sort of invertebrates. We have millipedes, which are macrodetritivores. Basically, they have these chewing mouth parts, and so a lot of them actually can take what you see in the litter here, break it down into smaller chunks that then allow for bacteria and fungi to complete the decomposition process. We have harvestmen. Some of them are scavengers. Some of them are predators. We have a group that's really, close to my heart, which are spiders. They're sort of the top predators in the soil ecosystem. They eat other invertebrates that keep their populations down. We have gastropods, the slugs. Some of them are detritivores. Some of them are herbivores. Another really dear organism here would be springtails. These are incredible organisms that a lot of people don't even know exist, but they are really small, close to microscopic. But one of the things that's really important about this group here, for one, they look like cartoons under the microscope, very adorable. But they actually serve as sort of microdetritivores, so they can break down some of the complex organic matter to smaller chunks, but they also, a lot of them feed on fungi, which can actually stimulate fungal growth in some contexts. And so they have some really important interactions with fungi. And so also very incredibly abundant and diverse, depending on the system, but incredible organisms that have an important role in the soil ecosystem. Another group are mites. Mites are incredibly diverse. You have predators, you have fungivores, bacteriivores. I mean, they do just about everything. They also incredibly hard to identify, partly why they're so understudied, but this is another really important group that occurs in the soil. We have wood lice or roly-polies, depending on where you are in the States, you have a different name for them. But another sort of macro detritivore, so they have grazing mouth parts that can break down organic matter. We have worms like earthworms and so forth. And so, and as I said, this is just a subset. You have archaea, you have nematodes, you have tardigrades. So it's a whole host of different organisms that exist in the soil, and they all have a function in some kind of way that really can dictate how nutrients are recycled, that then allow for what we know as our terrestrial system to really exist as they are. And so these groups are just a subset, but you can envision that they all have an important role in how these systems function in that case there. With that in mind, I kind of sort of alluded to all the different groups that occur, but there is some structure that we kind of think about. And so we kind of think of it as a sort of a bottom-up control community, meaning that as you sort of increase resources going into the community, all the different sort of organisms and trophic levels also increase with respective changes in how nutrients are recycled. So just to kind of walk through what this looks like here. So you have your typical deciduous forest. What happens is that they drop a lot of organic matter, so thinking about leaf litter and so forth. It kind of serves as the sort of organic matter pool as shown here. As you get sort of more organisms, more organic matter, you get a stimulation of what I call microflora. So in particular, this is bacteria and fungi. What happens is that as you get more organic matter, you get more bacteria and fungi, and their activities kick up, and that's going to release nutrients into the environment that then really impact the plant community. As you get more macroflora, that also stimulate primary consumers. So thinking about springtails in this case, or batid mites and other sort of nematodes in this case that are bacteria and fungivores, they all sort of benefit from that in this case. And then we also have influences on how nutrients are recycled in that case here. And so we also think about, they have predators. So these primary consumers have predators such as centipedes, certain species of, well, most species of spiders in this case. We also have nematodes that can be predators. We can also think about mite, so mesostigmatic mites are also predators, and their interactions can really influence how nutrients are released from primary consumers in this case here. So what I mentioned is that this being sort of a bottom-up control system. What we see is that as you sort of decrease resources going into the sort of soil food web, you see sort of shrinking of all of these trophic levels, so the microflora, primary consumers, predators decrease, and that should, in theory, slow down nutrient recycling in the system here. The same could be true on the contrary. So as you sort of add more resources, you get a bigger pool of microflora, primary consumers, and predators, all with this idea that they accelerate how nutrients are recycled in this case here. And so this is kind of a model framework that we work with in the soil food web. Just as a hint, this is an incredibly oversimplification of what really happens in the soil. But part of my work is really to try to unravel that complexity, to really understand how changes in the soil and terrestrial system can really impact how the food web is structured, and then what are sort of the consequences for what happens above ground. And so my work is really to try to unravel that complexity because it is very, very complex, but it also has really important impacts on how these systems are structured. So with that in mind, we know we have complex system, but you can envision that changes in the environment will probably have a pretty unique signature on how the soil food web is structured, and then how that can really impact how ecosystem functioning occurs, but then also how nutrients are recycled more broadly. So one can think about, deforestation or sort of habitat destruction. You can envision that when this happens, that's going to remove critical resources from that environment. And because we believe this a bottom-up control system, one can envision that in this case, that's going to drastically shift the structure of that food web, and then it's going to have really important consequences for how nutrients are recycled. We also can think about invasive plants. These are sort of organisms that can get into systems and completely dominate the plant community, and that can also change the composition and quantity of resources that go into the soil ecosystem, with really important consequences for how nutrients are recycled. We can also think about pollution or nitrogen deposition, all forms of like fertilizer inputs and so forth that could really change the quantity and type of resources that go into the soil food web, with really important consequences for what type of plant community that you may have in that case there. So as I mentioned on the previous slide, we have a framework to work from, but the reality, we don't really have a good grasp of how these different factors can really change the structure of these soil ecosystems, and how does that sort of relate to ecosystem function in that case there. And because we know that humans are drastically changing the environment, we're doing things that we've never done before, and it's accelerating as the population growth continues. It's really important for us to really understand how resource changes as a function of global change. How does that sort of relate to the soil ecosystem, and what are the sort of consequences for ecosystem function in that case there? So with that in mind, I'm gonna have two of my favorite case studies that I'll be presenting from my past work. With the first one, we'll be looking at a case study of famine. So what happens when you have an invasive plant that comes into a system that erodes the microbial community, and what are sort of the downstream effects on the soil food web? And then I have arguably my favorite case study, which is looking at sort of how resource addition through resource subsidies can really actually transform an ecosystem. And this was take me to some of my work I've done in Iceland. So with the first case study, I'll be looking at how garlic mustard can really change the structure of soil food webs by actually removing or sort of eroding the diversity and abundance of fungi in this case here. But this kind of takes me to an entirely different subdiscipline of ecology, which is invasive plant ecology. And so I will kind of have some examples of, for one, why they really matter in the first place, but then thinking about how they can actually change soil food web structure in this case here. So invasive plants, generally speaking, erode the diversity of the systems that they invade. Oftentimes they originate from another location, another continent, and they are introduced into a novel ecosystem. And because they don't actually have predators or herbivores in this case here, and And for a variety of other reasons, they may have quicker adaptation to the local environment. They become overwhelmingly competitive, and they can actually erode the diversity of that system by basically outcompeting all the native species in this case here. So we have some really classic ones, so like Japanese knotweed in this case here. They can really invade and just become overly abundant and push out all the native vegetation. As a little hint here, this is not what it's supposed to look like here. It consumes this little girl, and it has this amount of biomass that is really hard to kind of fathom when you think about an invasive plant coming in. We have invasive Phragmites, another plant that invades wetland ecosystems, can drastically change the structure of that ecosystem, pushing out the native plants, and also can reshift what the food web looks like. We have common buckthorn that can occur in the upper Midwest. This is also one of my least favorite invasives because, as the name suggests, it has thorns, and so they create these very dense thickets that is almost impossible to walk through. And 5 feet can feel like 50 feet when you're in one of these thickets in this case here. And something that we actually now have in Texas is kudzu, which is this invasive vine that comes in and basically just coats the entire vegetation. And one can envision that that's really pushing out all the native plants in this case here. So typically, studies have shown that when you get invasive plants in a particular system, they usually push out the native plants and can drastically change the biomass and the composition of that ecosystem. One can envision, though, that these individual plants that come in, as they sort of invade, they're going to change the type of resources that will ultimately go into the soil food web, and that can actually have some really important impacts on the system in that way. So for instance, as I just talked about, the common buckthorn has these very nutritious leaf litter, and so what typically happens is that once the leaf litter drops in these dense thickets, it actually accelerates decomposition faster than like an oak or maple litter in this case here. And so what happens is that in a system that should have very dense leaf litter layer, it accelerates decomposition in such a way that it's now bare ground. And one can envision that that soil ecosystem is not going to function in a natural way and will have some really important consequences for how nutrients are recycled. Similarly, you may have some plants that are more resistant to fire or less resistant to fire, so they can change disturbance regimes. Just the sheer abundance sometimes can change the microhabitat. You may have more shade or crowdedness. In some cases, that can change the amount of moisture you may find in the soil, which are incredibly important for a number of invertebrates as well. And then you have some invasive plants like black locust that can actually change the soil chemistry. And because a lot of these organisms that live in the soil are very sensitive to these changes, one can envision that certain invasive plants can really change what happens below ground from these various components. One of the sort of first studies I was involved in was really trying to understand, well, at a broad level, we know that these invasive plants could theoretically have a negative impact on certain trophic levels of these ecosystems. And so I kind of undertook a sort of global sort of meta-analysis and basically summarized all the data at that time that was available that were looking at the impacts of invasive plants on certain trophic levels, both above ground, as shown in green, and below ground, as shown in brown, with the idea being that, at least at this time, we thought that maybe the green food web would be more sensitive to invasive plants than the brown food web, given that the green plant tissue would actually have a more direct effect on the structure of the food web, whereas the brown food web, things that live in the soil, will be a little bit more buffered because you have litter that drops, and that still has the similar structure of that ecosystem. Needless to say, what we've actually found is that depending on the type of ecosystem, you do see some pretty pronounced impacts. And so what we found in wetland ecosystems, for instance, we found that primary, secondary consumers in a green food web, so thinking about herbivores above ground, grasshoppers, leafhoppers, and so forth, seems to be drastically negatively impacted, whereas our sort of predators, so thinking about web-building spiders, for instance, actually seem to be positively impacted. We didn't see any impact on issues with the brown food web, both for the primary and secondary consumer. In woodland ecosystems, what we found, similar to what we found in wetlands, is that the primary consumers were negatively impacted, but we found some really interesting impacts below ground in woodland ecosystems where we saw that primary consumers in the soil food web, such as springtails or batted mites and so forth, they seem to decrease, whereas the predators also seem to increase in this case here. And so we didn't see anything at all in grasslands. It seemed like there were really no strong impacts of invasive plants on those particular trophic levels. But this was particularly interesting to me from a below ground perspective. And so it sort of merited further investigation. Okay, what is really going on below ground that you may see a negative response of like springtails and or batted mites, whereas the predators seem to increase in this case here. One of the issues with a meta-analysis, though, is kind of just summarizing large data that have been published. It's not really mechanistic, but you can kind of see these really broad patterns that can at least point you in the right direction. But because it's not mechanistic, it kind of gave me some motivation to actually try to unravel this more with observational and experimental studies to kind of get at that more closely. In comes garlic mustard. Garlic mustard is this very interesting plant. It was brought over approximately in the 1860s, mostly brought over as a herb. Garlic mustard is where you get the garlic mustard sauce is where this plant is where this originates from. And it was brought over over the East Coast and has now been around for a while, but it is this really interesting invasive plant. It has some unique features that was the sort of focus of my work in this case here. So it's a biennial plant, meaning it has a 2-year life cycle. In its first year, it has these very small, inconspicuous sort of rosettes. It then kind of bolts pretty early on in the second year, usually around April or May. It then goes into senescence probably around July. And what's really fascinating that each one of these plants can produce up to like 5,000 seeds that can stay viable in the seed bank for up to 10 years. But the most interesting part is really in the second year senescence phase where it goes into senescence, the cell walls break open, and it basically releases glucosinolates, which are known to be antifungal. And it releases this all in the sort of surrounding root of this particular plant. And what's really fascinating, because it's antifungal, it basically has been shown to erode the diversity and abundance of fungi in the soil. What's really fascinating is that this actually is one of the sort of prevailing hypotheses around why garlic mustard is such a rampant invader, because most plants associated with mycorrhizal fungi, what happens is when you get garlic mustard, it comes in, it sort of suppresses the growth of fungi. It theoretically then removes the mutualism for the native plants, and then it becomes this sort of better competitor and kind of outcompete these native plants in this case here. But as I alluded to, that fungi also is an important food source for the soil food web, and one can envision that's going to have a really important impact on the soil community in this case. So this is a sort of healthy sort of forest that you would see in the Chicagoland area where this work was kind of conducted. And you can kind of see in the background, there's some leaf litter layer there. There's really no dominance of a particular plant species. And it seems to be fairly healthy for a fragmented forest in the Chicagoland area. This is what you can get with a garlic mustard invasion, where it just basically coats the understory. And you can envision that if each one of these plants will be producing glucosinolates that is antifungal, one can envision that it's going to really drastically change the fungal community in ways that can alter what lives in the soil, what feeds on the fungi, and ultimately restructure that soil food web in this case here. So this prompted some studies that I've conducted to really try to understand what happens when you do have garlic mustard invasion. And so going back to this example, so you have native plants, what happens is that they sort of release organic matter into the soil environment. That has a positive influence, in this case, fungi, so soil fungi, mycorrhizal fungi, it will be beneficial for them. That then sort of has a positive influence on the fungivores, that then stimulates more predators in the system here. What happens is that when you have a garlic mustard invasion, there's a lot of work that have demonstrated that garlic mustard negatively impact the growth and abundance of native plants. A lot of work that also demonstrated that it erodes the diversity and abundance of mycorrhizal fungi. And so one should expect then that garlic mustard will have a negative impact on fungivores and predators by this reduction of soil fungi. So effectively, as you remove soil fungi, all the fungivores and their associated predators will have to decrease because you have this removal of fungi in the system in this case here. So to kind of test this, I actually conducted a slate of different studies. I'll kind of present one that had the more clean results, but effectively what we did is we ran an observational study looking at natural populations of garlic mustard. And within each one of these sites, we were particularly interested in studying how these garlic mustard patches compare to what we call uninvaded patches. So we had patches that were 1 by 1 meter in size, and we compared it to an area that was roughly not too far away from that invaded patch to kind of see how that entire community looked in terms of both the plants, the soil, all the invertebrates below ground. And so across these 5 sites, we had 10 sites per site, and we had 50 plots total, and this was a 2-year study. And as I mentioned, we looked at everything in these particular plots from the vegetation. We also looked at the mycorrhizal fungi in multiple ways. So we did a washing technique to actually measure how much hyphae occurred in the soil. We also did some next generation sequencing to look at the DNA structure or the community of these fungi. And we also looked at all the arthropods that are associated with each one of these patches in this case here, all with the idea we'll be able to sort of relate how garlic mustard invaded patches compared to those uninvaded patches. I'm looking at the entire soil food web in this case here. So at just a very coarse snapshot, what we found is that you do get a huge increase in mycorrhizal hyphae when you are looking at the control versus the invaded plots. And you can see that's a pretty strong signal in this case here. So these sort of represent sort of standard errors in this case here. Control is the uninvaded, and then the invaded is the garlic mustard invaded plots here. And so what we found is that there was a huge difference between the invaded and uninvaded plots. When just looking at the fungivores, we saw a similar sort of pattern, albeit it wasn't as strong as the mycorrhizal hyphae in this case here, where we found more fungivores associated with the control plots as opposed to the invaded plots. When we looked at the predators, we actually didn't see a strong signal, at least in this very coarse measure, but there is a little bit of a signal that shows that there is a higher abundance of predators in the control plots relative to the invaded plots in this case here. I will say this is probably not the best sort of way to really look at this interaction, mainly because this is not controlled for how dense the garlic mustard patches were, or thinking about how they occurred over the 2 years. We really didn't account for that in this analysis here. And so I'll be showing some other ways in which we really try to get at this more specifically. One of which is looking at the fungal communities. And what we have here is sort of garlic mustard invaded patches compared to the uninvaded patches. And this is like representing the fungal community. So each one of these symbols represent a fungal community. So, and how you read this is that the closer these plots are, we represent very similar communities of fungi, and the further apart these are, represent very dissimilar communities. And so what we found is that we basically saw that there are entirely different communities of fungi. And what we found is like the most abundant fungivore in our data set, or batid mites, tend to actually correlate with the uninvaded plots more than the invaded plots. And then this direction shows that there's usually a higher abundance with these associated plots in this case here. So this is one of the 5 sites that we looked at. This one showed the strongest signal, but effectively we saw a similar pattern across all 5 of our sites here, which is that you get very different fungal communities, and they tend to always correlate with our most abundant fungivore in this case here, suggesting that the fungal community is really driving these differences in the arthropod community that's a function of whether garlic mustard was there or not. We also did a structural equation model, which I would argue was actually the best way to really look at how those interactions occurred across all those trophic levels. It accounts for the partial correlation coefficients, and you can kind of factor in site, location, and year. And so it allows for you to kind of tease all those apart to really understand how each one of these trophic levels respond to one another in this case here. And so you can kind of read it as a correlation coefficient. So the closer that number is to 1 represent a very strong pattern, and also had increased the width of these arrows to kind of indicate the strength of that sort of response in this case here. So with that being said, we found that across 2 years and across all 50 plots, we found that native plants had a positive relationship with microflora, in this case fungi. So the more native plants we found is a positive response there. This was not statistically significant, but at least it's in the right direction. However, we did find that as you have more fungi, we found a very strong positive interaction between microflora and fungi and fungivores, I should say. And similarly, as you get more fungivores, we found there was a really strong really strong positive relationship between fungivores and predators in this case here. As we looked at the impact of garlic mustard, no surprise, our design was sort of designed to do this, but we found that as you get more garlic mustard, you get a strong negative impact on native plants. We also saw a very strong negative influence on garlic mustard on microflora. And then we see these indirect effects that are negative for fungivores and predators in this case here. I know these may look like they're small values, but I actually think this is underrepresentation of actually what the impacts are, namely because the way that we measured fungi, we couldn't really detect between living and dead fungi, and so it's kind of a coarse measure. But even then, we were able to see that garlic mustard had a negative impact on fungivores and predators indirectly through this reduction of fungi in this system here. So with that in mind, I'm kind of really fixating on one particular trait of an invasive plant. Admittedly, garlic mustard is this very unique plant because it actually can suppress fungi in ways that other invasive plants can't. And so this kind of begs the question then is like, well, is it unique about garlic mustard in terms of the impact on soil communities? Or is it something that is more general in terms of how invasive plant root traits can really impact soil communities? And so this kind of brought me into a sort of recent collaboration I had with one of my colleagues at the University of Illinois at Chicago. She's a macroecologist. And so we were really particularly interested in is if we can kind of compile a data set across the United States to see if in fact root traits are different between native plants and invasive plants. And if so, do they have a unique signature on how they impact the soil communities? In this case, we were looking at microbes in this case. So basically, we compiled this data set that was really large. So we had a continental scale data, came from Neon. So we had data for native plant cover, invasive plant cover. We had root trait data from the TRI database and others. And we also have microbial composition, looking at phospholipid fatty acid analyses. So by compiling these data, it took a long time to do this. We were then able to kind of look to see if invasive plants have different sort of root traits. How does that sort of relate to plant cover? And then what does that look for? How does it look in terms of the composition of the microbes in this case here? And so all in all, we had 162 sampling plots from 94 invasive plant species across the United States in this case here. And we had data across 5 years. So very quickly, all this to show is that we looked at a suite of, I think, 9 traits. And what we found is that the specific root length of the invasive plants seemed to be higher than the native plants. So specific root length, we think about the fine roots that plants can have. A lot of those were much higher for the invasive plants compared to the native plants. And typically that means that they are acquiring nutrients faster and they grow much more quickly with the possibility of breaking down. And interestingly, we found that root tissue density was actually higher for the native plants, suggesting they have a little bit more recalcitrant root traits for the natives compared to the invasive plants in this case here. We looked at other sort of traits in this case. We didn't see strong differences across all of these particular plots in this case, but we did see a pretty strong signal for specific root length being higher for natives, and then the root tissue density was much lower for invasive plants in this case here. What was really truly fascinating about this, though, is that when we actually looked at how they sort of related to the microbial community, what we found is that in these heavily invaded plots across the United States, regardless of the ecosystem, regardless of where they were from a sort of latitude perspective, we saw that these heavily invaded plots had basically the same microbial community, whereas in the uninvaded plots, we found that there were such more variation across those represent the microbial community. And what we found is that, these highly invaded plots were very different to the non-invaded plots, and even moderately invaded was very different than the non-invaded, suggesting that as you get these high amounts of plant cover, invasive plant cover, it actually erodes the composition and the diversity of those microbial communities. And this was striking to see across the entire continental US in this case. So take-home message for this first segment is we basically saw that plant invasions can have pronounced impacts on soil communities. The changes in food web structure in the garlic mustard example seems to be related to fungal resources. And one of the things I think is really important to think about is that this can result in changes in the native plant abundance, but also is certainly sort of related to the traits associated below ground with these invasive plants in this case here. And so one can envision that the way that plant communities can be impacted by invasive plants can be a function of both its abundance, but also things that are occurring below ground. So with that said, with the time left, I will kind of pivot to one of my sort of favorite study systems, which is this pretty unique lake in Northeast Iceland. And we'll be looking at the other tail, where we're looking at what happens when you add a lot of resources to the ecosystem in this case here. One of the things that I have to sort of prep, though, is thinking about, well, this entirely different system of subliterature, which is thinking about ecosystems as being part of a mosaic in a landscape. And so we typically think of ecosystems as being bounded, but in reality, they are sort of a mosaic of interacting ecosystems in a landscape. So just to kind of provide an example, we have sort of a riparian forest right next to sort of river in this case here. One can envision that there is going to be a lot of litter and sort of nutrient inputs that can go right into the river, that can stimulate algal production in the river, that can sort of support aquatic insects and fish and so forth. Some of these aquatic insects and fish can actually end up dying on the landscape and can actually fertilize this riparian forest. And so one can see how they can sort of relate to one another in a really meaningful way, and in particular at the interface in this case here. So one of the cool examples of this sort of flow of nutrients across the ecosystem is the sockeye salmon. Probably occur not too far from this particular region here, but typically they spend their entire life out in the open ocean. As they sort of trying to migrate up to their sort of spawning grounds, they run into a whole host of predators, such as grizzlies, other sort of predaceous birds and so forth. A lot of the sort of subdominant grizzlies can retreat under the sort of riparian forest, and basically they can deposit those carcasses on the landscape, and as they sort of decompose, they can release all type of nutrients into a riparian forest, such as carbon, nitrogen, and other macronutrients, phosphorus, and so forth. And what happens is that those nutrients can actually bolster the primary productivity of that riparian forest in really meaningful ways that can reinforce the riverbed, can help with the flow of the river, that then allows for the sockeye salmon spawning grounds to be more productive. And so as you get sort of the death of the carcasses, the death of these sockeye salmon, releasing nutrients, it bolsters the riparian forest, that then allows for the river to have the perfect spawning grounds, can then stimulate sockeye salmon. And so you can see how the interaction between the aquatic and the terrestrial side of things can have a really important influence on how these ecosystems are structured. So this brings me to Northeast Iceland, which I've done some work for 3 consecutive summers and was kind of looking at similar sort of ideas, but in a very different context in this case, which is Lake Mevat. Lake Mevat is this really interesting lake. It actually translates to midge lake, and it's this really unique lake that produces huge numbers of midges. And those midges are thought to actually have a really important influence on the terrestrial landscape that's surrounding it in this case here. But a little bit of sort of context around Lake Mevat is this really nutrient-rich lake. It has like all type of springs that kind of pump in from the surrounding landscape, such as pumping in nitrogen and phosphorus and so forth. It's fairly large. It's also very shallow. So at this latitude in Iceland, you basically get 24 hours of sunlight in the summertime. And so a lot of that sunlight can penetrate the water column and make it down to the bottom of the lake and the benthos that can stimulate detritus and algal growth and so forth. And it becomes this very, very productive lake. So much so, there's this one species of midge that just kind of takes off called Tannitarchus gracilensis. And basically what this species does is that the male and the females, they sort of congregate and mate on the terrestrial landscape around here. And the females return to the lake, drop their eggs. We actually don't know what happened with the females at that point, but the eggs sort of hatch and then they kind of go through a series of instars. They all pupate as adults and they then migrate back across the terrestrial landscape here. What's really fascinating about this area of Mevat, it should be low-lying shrubs like heathland vegetation. But as you can kind of see here, we get some really productive grasslands that occur around here. And so there were some thoughts that maybe it's the midges and their activities that actually can stimulate the productivity that we see here. And so what happens is that when these midges emerge, they create these huge mating swarms that can have really pronounced impacts on the terrestrial landscape, then that can kind of fertilize what happens around the lake in this case here. So to kind of demonstrate, this is kind of what happens during one of the midge swarms. This is usually around late May, early June. You get these large numbers of midges, sometime in the trillions that can kind of emerge. And each one of these are like specks of midges flying around. And so when we did some back calculations to kind of estimate how much biomass is coming out of the lake, it would be equivalent to like a humpback whale being slapped on the landscape in the first 50 meters. So that's how much biomass is coming out of the lake on a given year here. And as you're sort of driving around Lake Mevat, you get these clouds that you can see. You literally think they're clouds, but these are just actually midge swarms. And so as I mentioned that the males and the females, they mate. The males just kind of hang out. They just kind of chill out in the landscape. We don't know what happens with the females, but what happens is that the males just effectively die. And what is really shown here is actually one of my favorite photos of all time. This is one of our undergrad interns that was working with us one summer. Karen is like 6'1", she's very athletic, got these large hands. What you see here is that half of her index finger is submerged in midge carcasses. And so one can fathom that these midges have a lot of carbon, a lot of nitrogen, a lot of other macronutrients. You can envision that as they decompose, they are effectively just fertilizing that landscape. And so this was one of my favorite moments to kind of see that. Kind of gross at the same time, I admit, yeah. So with that in mind, so kind of set up some studies to kind of really look at what happens when you have midges and how does it actually transform that heathland into a grassland. And so I kind of conducted some experiments. I'll kind of present one of my favorite ones to kind of really illustrate that. So going along with the model here, so we have a typical heathland. That's typically what you should expect in this area of Iceland. What happens is that when you add midges to the landscape, it really just transformed from a heathland to a grassland. We have some studies that kind of show that there. We also have some studies that show in the both above ground, below ground, you get increases in primary consumers and predators. But what we didn't know is like what happens with the microbes and how does that sort of relate to the nutrients being recycled in this case here. And so as a part of this study, I was really honing in on the microbes and then how does that sort of relate to nutrient cycling. So to do this, I did a very clever but elegant experiment, which is looking at this midget digital experiment. So basically there is this lake that's not too far from Lake Meevat, maybe less than 6 kilometers. But for whatever reason, this lake is roughly the same size as Meevat, but it didn't have any midgets at all. And we basically ran this midget addition experiment where we back calculated what would happen at Lake Meevat and put midgets on a landscape in a similar amount to kind of see what happens when you do that. So this was basically a simple one-level experiment. We did midgets versus no midgets. It was a site that had low midget abundance, so we can kind of rule that out as a confounding variable in this case. And we had actually very small plots. We added about 100 grams of midge carcasses across that to kind of sort of reflect that amount of biomass that would be typically coming out of Lake Meevat. And we basically measured the entire ecosystem from plant composition. We also looked at litter decomposition. And this was kind of one of the neat things, I think. So we took like actual plant litter, put them in the litter bags, and then we just added midges to see how it actually increased or decreased litter decomposition. We also did some soil cores where we took the soil, did some incubation experiments to see how much carbon respired. And all with this idea to really try to understand how these midges were impacting. We also looked at phospholipid fatty acid analyses as well to look at the microbial community. And so we kind of looked at pretty much the entire, albeit very small, but we looked at the entire ecosystem in this case here. So just after 2 years, we were able to recover that pattern. So over here, we got no midges. It's kind of a typical heathland plot. Over here, we have a midge plot, and you can just see the grasses just kind of take over and dominate this particular ecosystem here. You don't even need stats to kind of look at that, but that was basically across all the plots in this case here. And what was also really fascinating is just by adding midges, you increase litter decomposition by 2.5 times. So as I mentioned, this is not the decomposition of the midge carcasses themselves, but just by adding midges to the plots, it's actually speeding up how fast litter is decomposing in this plot in this case here. So it rapidly intensifies the amount of nutrient cycling happening in these plots here. There is certainly some variation across this as a box plot. These are sort of medians here, but one can see that there's a pretty big difference there. And no surprise, what we found is that in our incubation experiments, where we measure how much carbon was being respired over 30 days, roughly, we found that there was a 60% increase in microbial activity, really showing that it's the microbes that were really responsible for breaking down those midge carcasses and sort of liberating all of these nutrients that then allow for these graminoids or grasses to take off. One thing I should say, these graminoids are fast growing and they're probably better able to sort of adapt or be able to respond to changes in nutrients, whereas the heathland vegetation, like the shrubs, are slow growing. They just get outcompeted by the grasses when you have this liberation of nutrients. Also, when we looked at phospholipid fatty acid analyses, we found that the microbial abundance was much higher, both fungi and bacteria in these midge addition plots compared to the control plots. So kind of when we put it all together, what we find is that, midges transform the heathland to a grassland. You get an abundance of primary consumers and predators, but we also get an increase in the microflora, both bacteria and fungi, by 1.5 times. We also see nutrients are released and sort of recycled 2.5 times faster when you add midges to the system in this case here. Last but not least, this is kind of some ongoing work, but we wanted to kind of do some modeling of those dynamics. So we were trying to look at sort of carbon mineralization. So we were doing some energetic modeling to kind of tease that apart here. This is kind of a busy slide, but I think this is kind of one that I was just very excited to show. These are just basically the different components of the ecosystem, both above ground and below ground. These are kind of simulating sort of carbon mineralization across these different trophic levels. And what you can see here, this is just like your typical Heathland plot when you don't add midges. And so you don't really see a whole lot going on in this case here. But this is just how complex and how much carbon is being recycled when you have a midge addition plot here. And so you can see that the entire soil food web gets more complex, more carbon is being cycled. We actually get organisms that weren't actually here. Once there's some organisms that weren't actually here, that's now over here when you added midges. And so it actually shows that in this particular case, the midges are actually stimulating the entire ecosystem in really meaningful ways. And so one can envision then in the context of global change, if you were to decouple what midges are doing on the landscape, it can actually have really negative consequences on that terrestrial landscape. So to summarize for time in this case, I have a couple of just points here. So all this to say is that disturbances affecting soil community structure and function will have really important consequences for the things you see above ground. So you can think about this, albeit a unique case, but you can envision that cases where you have resources coming in this case, insect carcasses can really accelerate decomposition rate. It can shift plant composition in this case, and also really stimulate microbial activity, which seems to be the one that's really responsible for shifting the nutrients in ways that allow for the plant composition to shift. On the contrary, you can have a situation where if you kind of altering the soil microbial community, in this case through allelopathy or invasive plants more broadly, that can have really important impacts on soil food web structure. We saw a very strong signature of fungi and fungivores being reduced. Predators seem to be indirectly affected, primarily through this reduction of fungivores in this case here. And if you don't learn anything from my talk at all, one of the things I just want you to kind of walk away from is that the soil food web is incredibly important for recycling resources and really important for structuring the ecosystems as we know them above ground. So whenever you sort of look at your really neat ecosystems above ground, just know that it's really the soil food web that's responsible for that. So with that being said, I would acknowledge all the great people that was associated with this project. And just for the sake of time, I just would be more than happy to jump into questions. Any questions? Go there first.
That include the carbon that's stored in the biomass itself, or is that completely separate different carbon that can produce?
That's a great question. So the question is, going along with this figure I have here, is that is this showing just the amount of carbon that's going in there through the midges, or is it changes in what the midges are doing that then impact the rest of the soil food web in this case here? That's a phenomenal question. And actually, if you were to sort of calculate here, what you see is more carbon being cycled through this midge addition plot than the amount of carbon we actually added in the midge carcasses. So basically, they're stimulating a lot more activity in ways that's like liberating more carbon than otherwise would be in this case here. That was really fascinating to me to see that like actually when you add midges, it stimulates a lot more carbon release than it would be otherwise. But yeah, that's a fantastic question. Thank you. Yep. Most of them have fish in there, including Lake Meebot. Yep. Yep. That's a good question. Lake Meebot is fairly unique in that it has a pretty diverse community of fish where the site that I actually ran this experiment, the fish community is not as abundant or diverse in that case there. I don't really understand exactly how that happened in this case. And in fact, no one really knows, but it's just a really fascinating part of like this part of Iceland that there's just some unique features about these lakes that we fully don't understand why they are different. But part of the work we try to do is like, how did these midges actually get there? Why are they so abundant? Because they're not like that anywhere else in that on the island of Iceland. Any other questions? I go there first, yeah.
rid of the, like, fungal and like,
Yeah, no, that's another really great question. So one of the things about garlic mustard and the whole family, Brassica, they basically are non-mycorrhizal. So the entire family don't associate with mycorrhizal fungi. I don't know exactly how they're able to compete with other plants that, well, I mean, obviously they take away that mutualism, but for whatever reason, garlic mustard doesn't associate with mycorrhizal fungi. That being said, there's certainly some evidence to show that in these long-standing populations of garlic mustard, they do thin out after a while. It takes a long time, but it could be a buildup of pathogens or just the fact that when you have these long-standing patches of garlic mustard, that they start to get outcompeted by the native plants. But it takes a really, really long time to get to that point. And usually in the first 50 years, they can be super hard to get rid of, for sure. Yep.
So I'm actually interested in this whole grassland transition. I'm not a soil person, so I'm probably prone to misunderstanding, but I'm almost curious. So you have a net more carbon in the, like, there's more carbon in the system with the midge addition.
Right.
It's not just the midges. I'm almost curious
Mm-hmm.
in the grassland system, if you're excluding midges, so rather than in the system where you added midges and you saw that transition, you go in exclusionary. Do you think you'd see that grassland, like that patch persist as a grassland, and maybe it's kind of gone through a new, like it's self-sufficient as carbon, or do you think it will revert back pretty quickly to that?
Yeah. Yeah. Heathland. Yeah. No, thank you for that question. Yeah, that's actually, so this is just like one of the experiments. I actually also did a midge exclusion experiment at Lake Mevat where we put up these big cages, relatively big. It felt like it at the time anyway, but basically excluded all these midges from coming in. And basically it sustains for a long time. I also would say that my postdoc advisor put out these long-standing garlic mustard midge addition plots. And after sort of 7 years, he basically stopped adding midges and then kind of tracked how long does that grass effect last for. And it lasts a long time. And in fact, as they go back, those plots are still grassland plots. So once it like transitions to a grassland, it seems like it's very hard to get it back to a heathland plot. So it seems to be kind of a long standing pattern in this case here. Any other questions? Yes. Mm-hmm.
they're like anecdotes, and I don't know if like it's anecdotes judgmental or are they like are they good or are they bad? And bad, even, you know, like a.
Yeah. Now that is a good question. I would say that probably would be context dependent. For one, it kind of depends on the species events. So we got the red imported fire ant down in Texas, and we try and get rid of that every single time. But ants are incredibly important for turning over nutrients. So I would say if they are native and they're not really overly abundant, then they're probably serving the ecosystem really important services. And so I would be very hesitant to move them out, not unless they like some like pernicious invasive species. But generally speaking, they're most likely going to be good for the ecosystem.
For coming and spending for us some more time.